What if the real reason you feel exhausted, foggy, and slower after 40 has nothing to do with laziness or stress, and everything to do with tiny structures inside your cells that are literally dying off? I'm talking about your mitochondria, and what's happening to them after 40 is one of the most important stories in modern longevity science. Most people have never heard this explained clearly, so let's fix that right now. Your mitochondria are the power plants of your cells. Every single cell in your body, except red blood cells, contains hundreds to thousands of these microscopic structures, and their one primary job is to convert the food you eat and the oxygen you breathe into usable cellular energy called ATP. Your heart cells have about 5,000 mitochondria per cell because they never get a break. Your neurons have similar numbers. Now here's where it gets serious. After the age of 40, research published in the journal Cell Metabolism in 2013 by scientists at the Karolinska Institute in Sweden demonstrated that mitochondrial function declines measurably every single decade. By the time you're 50, you may have lost up to 30% of your peak mitochondrial efficiency. By 60, that number can climb to 45% in sedentary individuals. That's not a minor inconvenience. That is a fundamental collapse in your cellular energy infrastructure. And it explains almost every complaint people associate with aging: the fatigue, the brain fog, the slower recovery from exercise, the increased vulnerability to metabolic disease. So why does this happen? There are three primary mechanisms. First, mitochondrial DNA damage accumulates over time. Unlike your nuclear DNA, mitochondrial DNA has very limited repair mechanisms and sits extremely close to the reactive oxygen species that mitochondria generate as a byproduct of energy production. Think of it like a power plant that slowly poisons its own blueprints. Second, a process called mitophagy, which is your body's system for clearing out damaged mitochondria and replacing them with fresh ones, becomes less efficient with age. Harvard researcher David Sinclair, whose lab published landmark papers on this between 2013 and 2020, describes this as the cellular equivalent of never taking out the trash. Third, the signaling molecule NAD+, which mitochondria absolutely require to function, drops by roughly 50% between age 20 and age 50 according to research from Washington University in St. Louis. Without adequate NAD+, even healthy mitochondria cannot perform their core functions properly. Now here's where the science gets genuinely exciting, because none of this is irreversible. A 2017 study published in Cell by David Sinclair's group at Harvard Medical School showed that restoring NAD+ levels in aged mice actually reversed many markers of vascular and muscle aging within just weeks. The mitochondria didn't just stop declining, they actually rejuvenated. Human trials have since shown that NAD+ precursors like NMN and NR can raise cellular NAD+ levels significantly, with a 2020 clinical trial at Washington University showing a 60% increase in muscle NAD+ in older adults supplementing with NMN over 10 weeks. But supplements are just one piece. Exercise, specifically Zone 2 cardio, which is that moderate intensity pace where you can still hold a conversation, is the most potent known stimulus for mitochondrial biogenesis, the creation of brand new mitochondria. Research from the Mayo Clinic published in 2017 found that high-intensity interval training reversed age-related decline in mitochondrial capacity by 69% in adults over 65. 69%. That is not a modest effect. That is a transformation. Dietary strategies also matter enormously. Time-restricted eating, specifically compressing your eating window to 8 to 10 hours per day, activates AMPK and SRT1 pathways that directly stimulate mitophagy, clearing out damaged mitochondria. Research from the Salk Institute in California has demonstrated this effect consistently in both animal and early human studies. Cold exposure is another validated lever. Studies from the University of Maastricht showed that repeated cold water immersion increases mitochondrial density in brown adipose tissue and may have systemic effects on mitochondrial efficiency. Even specific foods matter. Urolithin A, a compound produced when your gut bacteria metabolize pomegranates and berries, has been shown in research published in Nature Metabolism in 2019 to directly stimulate mitophagy in human muscle tissue. So here is your practical starting point. Number 1. Commit to 4 sessions of Zone 2 cardio per week, 45 minutes each. This alone is transformative. Number 2. Compress your eating window to 10 hours. Start with that. Number 3. Consider getting your NAD+ levels tested through functional medicine and discuss whether precursor supplementation makes sense for you. Number 4. Eat more pomegranate, berries, and walnuts to support natural urolithin A production. Your mitochondria are not your destiny. They are your opportunity. The research is clear, the interventions are available, and the window to act is right now.